Okay, welcome back. And this will be the start of the last unit of study for this course. It's also the last lecture that I will be posting for this course, sort of bittersweet, but here we go. We're still on this topic of catabolism, the degradation of fuel molecules in order to extract energy from them for our own uses. And the classic fuel molecule we have used thus far has been glucose. So glucose here is the go-to energy source. It is the preferred fuel molecule for degradation to ultimately extract its chemical potential energy and synthesize ATP. But there are other molecules that the body will use in either the absence or along with glucose, one of which would be amino acids. Now, this is a very rare sort of occurrence when animal systems catabolize amino acids for their own uses. Typically, it only happens under starvation conditions when all other fuel molecules have already been used up. So we don't see that as much, and as such we won't be covering the catabolism of amino acids here. Really, the sidekick or the secondary for energy production in animals are fats. So fats, as we know, fats are a store of energy, but the fatty acids, which make up triglycerides or fats, can be thought of as a backup, secondary source of a fuel molecule for degradation to ATP in animal systems. So if fatty acids are the fuel molecule that is going to sort of this secondary fuel molecule to the preferred fuel molecule glucose, we typically think of fats as in an energy storage. We keep this energy storage system separate in order that we can access it if we run low on glucose or that sort of thing. The glucose, being the preferred energy source, is typically stored as glycogen, if we'll remember, mainly in our liver, but also in skeletal muscle. Fats, or rather fatty acids, also have a storage module, and that is with respect to triglycerides. Remember, our triglycerides, which we'll look at in a little bit more detail, were essentially stores of fatty acids. There's three fatty acids that are esterified to a glycerol, three-carbon glycerol molecule, to provide a neutral triglyceride, which are overwhelmingly considered fuel storage or energy storage. So these triglycerides are typically held, and we've seen this slide before, within these adipose cells, which are the cells of ultimately fat tissue. They're specialized cells, which the majority of their volume is really this storage container for triglycerides, and so that they just reside within there until they are called up and requested for if the body needs, or if animal systems need, additional energy. So we have to take these triglycerides and convert them, if you will, or hydrolyze them into their free fatty acids, and then these fatty acids themselves are shuttled into the mitochondria within a cell, and a different type of energy production system, which we'll talk about, is then recruited in order to form ATP. But to start, let's look at that process, or rather that journey, going from the triglycerides held within the adipose tissue into fatty acids, which can then be shuttled into the cells. So here's a representation or a structure of a triglyceride. It's a neutral triglyceride, and it is an energy storage unit. So we can see in here the linkage. There's that link, that glycerol linkage, holding together our three fatty acids in an ester form. So the first thing that ultimately happens if we require the freeing up of fatty acids. And we'll be looking at this primarily with the ingestion of dietary fats. So the triglyceride in question here would be considered something that's taken in as a dietary fat. And the first thing that ultimately has to happen is that enzymatically we get hydrolysis of those three ester bonds by an enzyme known as lipase. So lipase carries out, and typically this is within our digestive system, takes care of hydrolyzing those fatty acids. So now we have three fatty acids which can be then taken into our or uptaken into our intestines for delivery into the bloodstream to our cells. Going from triglyceride to free fatty acids can be problematic, however. Almost most of the nutrients and the foods we take in through our dietary intake can ultimately be dissolved in an aqueous medium. Our glucose and amino acids and that sort of thing are easily turned into are easily dissolvable, but triglycerides are oils, and oils tend to not dissolve in an aqueous medium. So we have to be able to atomize, if you will, or break down these sort of fat globules and from dietary fats within our systems, and we do this through the recruitment of bile salts. If you remember, bile salts were these steroid type structures that they're secreted into the digestive tract to help emulsify these fat droplets in the stomachs. I know that's very very appetizing around breakfast time or whatever, but here's a fat globule, or a representation of a fat globule within our digestive tract, and that has to be that has to be what's called emulsified, which basically means made much smaller, broken down into much smaller pieces. And here we are going from 1 to 3 pieces, but it would really from one fat globule to billions of them, very, very small. And in so doing, the more emulsified fat droplets that we get creates a much larger surface area overall on these droplets for the lipase and the bile salts to do their work. So it allows for a much quicker time period from going from triglycerides, which then can be acted upon by lipase through the action of bile salts, until we get to these free fatty acids. So these free fatty acids are then uptaken into the bloodstream and delivered to cells, which then make their way into the mitochondria of the cells and are degraded. They are catabolized in order to extract energy from them. This process is known as beta oxidation of fatty acids. And here is a representative long-chain fatty acid right here that we're somewhat familiar with. And we have to understand what beta oxidation is, we have to look a little closer at the structure of these fatty acids and the representation we put upon the carbons as they show themselves in distance from the carbonyl carbon, which is, as we know, right here. So let's open this up a bit. And what is beta oxidation? Well, on a fatty acid like we see here, one of the ways in which we can number or refer to the different carbons in a carboxylic acid, we've seen that in UPAC nomenclature, where we will go, the carbonyl carbon is the 1 carbon, the next carbon in the chain, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and so on. Well, there is a different way of doing this. And we've talked about this a little bit before when we talked about omega-3 and omega-6 fatty acids, is this Greek alphabet naming system where the carbonyl carbon stands alone. That's the carbonyl carbon. And the carbon adjacent to it is known as the alpha carbon. The carbon 2 carbons away from that carbonyl carbon is known as the beta carbon. And then we would have gamma carbon, delta carbon, epsilon carbon. Those are infrequently used. And we would get the way we looked at it before in talking about essential fatty acids was all the way to the other end of the molecule. This end carbon was the omega carbon. This is the last one, omega carbon. We counted in, whether 3 or 6 carbons on this to identify where the first double bond showed up. So this Greek numbering system is going to be important. Specifically, since alpha is the first letter of the Greek alphabet and beta is the second, is this beta carbon right here now, which is we're talking about when we look at what is beta oxidation. So that carbon, that beta carbon, ultimately is going to become oxidized in this energy harvesting process. And what we mean by that is we're going to decrease the number of CH bonds at that beta carbon, and we're going to increase the number of CO bonds at that beta carbon. We're going to see that ultimately that is what happens in beta oxidation. So all eyes now are going to be on this beta carbon as we follow it through this process. So let's, since we're only looking at that alpha and beta carbon on here, the rest of this fatty acid tail is kind of unnecessary. So what we'll do is we'll just abbreviate that away. So that we have an alpha carbon or beta carbon and all the rest, which we don't care about. We'll just write right here as fatty acid. Now, in the slide that we're going to be looking at that shows this process of beta oxidation, I'll show over here on the left-hand side what that's actually going to look like. It looks similar to this piece down on the bottom, but here now we have our carboxylate because we're at physiological pH, our alpha and our beta carbon given as condensed structures, CH2s, as opposed to carbons with bonds to hydrogens, and of course the rest of that structure given as just fatty acid. But this is the structure here over on the lower left that we're going to be following through this process. So this is going to end up being somewhat of a busy slide, but we're going to walk ourselves through the beta oxidation process. And in so doing, the biggest part of this beta oxidation is going to be very similar to something we've seen before. In that in nature, when nature settles on a process or a structure, something that works really well, we see that echoed throughout other types of processes, other types of systems, and so on. To start us out, over here in the upper left-hand side, we have that abbreviated fatty acid that we saw on the end of the last slide with our alpha and beta carbons. All eyes, remember, are going to be on that beta carbon. Now, similar to back in glycolysis and anaerobic respiration, we talked about 2 stages of glycolysis: the energy investment stage, where we have to burn already existing ATP to set up the fuel molecule for degradation. And then that is followed by the energy or payoff process, where we get more ATP out of it than we put in, so that we get a ATP profit. Similar with beta oxidation, we have this activation step. In this activation step, costs ATP. It is an energy investment, and it costs the equivalent, as we'll see, of 2 ATP. So that first step, that activation step, sets up our fatty acid in order to prepare it for beta oxidation. And all we're really doing is we're taking the fatty acid and we're making a coenzyme thioester out of it. So again, coenzyme A. Coenzyme A comes in and hooks up, if you will, through a thioester this fatty acid. What once was a carboxylate is now a thioester. So that is an important step. And it says here it costs 2 ATP. And I'm going to say that is equivalent of 2 ATP, because if we look at what is happening in that process, we get ATP is being converted to a MP plus 2 phosphates. PPI, it says there. That is 2 of the phosphates. So really, we're losing not 1 phosphate from ATP to become ADP, but 2 of them. So that one step, ATP to AMP, that's adenosine monophosphate, that costs the equivalent, since 2 phosphates are lost, the net equivalent of 2 ATP. That's why they call it 2 ATP when it looks like just one step. It's actually 2 phosphates net are being released from ATP to become AMP. All that aside, that is our activation step, that is our energy investment, and it costs the equivalent of 2 ATP. And now we have our template and we're ready to start. So we're going to put that template up here now so that we can start the beta oxidation process in which we're going to be harvesting energy. So here we go with beta oxidation. We have exactly what we had down here. We have our coenzyme esterified fatty acid. Let's remember, all eyes ultimately are going to be on this beta carbon right here, and it's going to be oxidized. So let's look at the first step in this beta oxidation process, in which, watch the alpha and beta carbons now, that we went from a carbon-carbon single bond to a carbon-carbon double bond. That's familiar. We saw that in the citric acid cycle, and I claimed that that was an oxidation. Why? Because we are going from total of 4 CH bonds to a total of 2 CH bonds. We've lost CH bonds, and thus that must be, by definition, an oxidation. Similar to the citric acid cycle and any oxidation, we cannot have an oxidation occurring without a concurrent reduction. And that concurrent reduction, in this case, is an FAD being reduced to an FADH2. Remember, beta oxidation, like the citric acid cycle, is happening inside the mitochondrion. Thus, any of these reducing equivalents, FADH2 or NADH, is a good thing, because we're going to go cash those in within the mitochondria for ATP by way of that oxidative phosphorylation that we saw at the end of the last unit. So here now we have our FADH2, which, as we see, can be then converted to 2 ATP. Remember that for every FADH2 converts to 2 ATP, every NADH, going back to NAD, drives the formation of 3 ATP. So now here we are with our carbon-carbon double bond, and let's see what happens next. Well, similar to before, we've seen this before. This carbon-carbon double bond allows water to add over it to form an alcohol. It's a hydration step. Again, so this hydration step now is not an oxidation or anything like that, but it sets us up for a final step in beta oxidation, in which this alcohol here is oxidized to a carbonyl. Well, we cannot have an oxidation without a concurrent reduction, and here we bring in NAD and we reduce it to NADH. That's a good thing in the mitochondrion, because that will drive the formation of 3 ATP. Let's take a closer look now at what we've done. Remember, this is called beta oxidation. Here's that beta carbon at the beginning of this, CH2. Here's that beta carbon at the end of all this, C double bond O. We've lost CH bonds and we've gained CO bonds. So again, as far as why this is named beta oxidation, beta means that beta carbon on what once was that fatty acid is now more oxidized. This fits with our idea that any fuel molecule being catabolized to extract energy from it is constantly bringing carbon to a higher oxidation state. And indeed, that's what has happened here. So let's look at what we have. This is sort of a weird molecule. This one down in the bottom, I'm going to put over on the upper right-hand side now. And we have the final step in here. And we have two carbonyl groups showing here, 1, 2. And let's take a look at what they are. Well, this first carbonyl group that we see right over on the right-hand side here, that was the original carbonyl of the fatty acid. This new carbonyl now, over to the left of it, that one was produced in the beta oxidation process from what once was a CH2 on that beta carbon. Now, here's where the magic ultimately happens. We recruit coenzyme A, will come in, another coenzyme A, will come in again and cleave this bond right here. It will cleave it. And coenzyme A will form a thioester with this new carbonyl, which leaves this somewhat familiar structure here. Now, if you just add a hydrogen to it, that is acetyl-CoA. Remember, we're in the mitochondrion. And acetyl-CoA can then be immediately power one turn of the citric acid cycle, which is ultimately, once all NADH and FADH2 equivalents are taken care of, 12 ATP. And look at what we have at the very end of things. We have that original fatty acid, which is esterified through a thioester to coenzyme A, yet it has been shortened by 2 carbons. 2 carbons. So this continues. Now this fatty acid, or this thioesterified coenzyme A fatty acid, which is now 2 carbons shorter, then goes back and starts the process again, only being 2 carbons shorter. And the process happens over and over and over, each time taking 2 carbons away from that fatty acid. Each cycle of that lowers or shortens that fatty acid by 2 carbons, by 2 carbons. It chews it up 2 carbons at a time until it's gone. So for each 2 carbons, we can take a rendering of this. Let me clean up this slide just a little bit here, and this is going to be important coming back through. Let's take a look. So for every 2 carbons on the fatty acid that is catabolized, let's do an ATP count. 2 there, 3 there, 12 there. So thus, as this fatty acid gets chewed down by 2 carbons at a time, it produces, or it allows to be produced, 17 ATP, 2 plus 3 plus 12. So let's take stock here. That's basically all of the players that we saw coming in sequentially in the last slide were taken from what I've got right here over on the left-hand side of beta oxidation, but shown in a more condensed structure. Here we have our activation step right here. Here we have our beta oxidation step right here with the thiolysis step right here, all put together. And as soon as I clear this up a little bit, we can see that going from here to here, we have shortened that original fatty acid by 2 carbons, and here again now we are showing the cyclic nature of it. That fatty acid, now shortened by 2 carbons, starts all over again and loses another 2 carbons, which that subsequent one, that is another 2 carbons shortened, goes back to the top, and we get this cyclic structure chewing up each time 2 carbons from that fatty acid. So I want to look at 3 specific reactions in here, because as I alluded to before, that has an echo to it, a ring of familiarity. And specifically, I'm going to bring in the citric acid cycle, again, which we saw in the last unit to compare this to. So here is the citric acid cycle that we talked about last time with our structure, our continually evolving or oxidizing carbon on the citric acid cycle. And I'm going to claim that the heart of the citric acid cycle is essentially what's going on in beta oxidation. So let's recruit exactly the reactions that we want to look at in both beta oxidation and the citric acid cycle. Now, in both cases, we have different directionality going here, which I'll put in here in the beta oxidation on the right. We have the series of reactions I would like to look at going downward by this arrow. And on the citric acid cycle, in this portion of it, I have it sort of swooping upward by that arrow right here. But let's look at both of these. And I'll start with beta oxidation over on the right. These three reactions involve going from a carbon-carbon single bond to a carbon-carbon double bond, followed by hydration of that double bond, followed by oxidation of that resulting alcohol to a carbonyl. And in so doing, we bring one FAD to FADH2 and one NAD to NADH, beta oxidation. Let's look over on the right. Here we have a carbon-carbon single bond being converted to a carbon-carbon double bond, just like we saw before with the concurrent reduction of FAD to FADH2. Carbon-carbon double bond being hydrated to form our alcohol, which we see right here. Alcohol being oxidized to a carbonyl compound with a concurrent reduction of NAD to NADH. They're essentially identical. Those three reactions in beta oxidation and the citric acid cycle drive the formation of one FADH2 and one NADH. Let's look a little bit closer at that on the next slide. So these are a little bit different renditions of beta oxidation over on the right and the citric acid cycle over on the left. But again, here we have it. Here's those alpha and beta carbons of the fatty acid, which those carbon-carbon single bond goes to carbon-carbon double bond. Similarly, down here on the bottom on the citric acid cycle on the left-hand side, single bond goes to double bond. In both cases, we are driving FAD to FADH2, FAD to FADH2. Back over on the right, that double bond is hydrated with water to form an alcohol. Over here on the left, that double bond is hydrated with water to form an alcohol. Back on the right, the bottom reaction, that alcohol is oxidized to a carbonyl group, driving the formation of one NADH. Over on the left-hand side, that alcohol is oxidized to a carbonyl, driving the formation of one NADH. It is literally identical, and that's the most important components of the citric acid cycle. It happens to be the exact same series of reactions, almost, at least in principle, as in beta oxidation. So in beta oxidation, we are losing 2 carbons at a time from that long chain fatty acid tail. We're snipping it 2 carbons at a time. So I'm referring to this in an unscientific fashion. Each of those, so we consider each run through beta oxidation, I'll consider as a snip. Very unscientific, but a snip means 2 carbons are snipped from that fatty acid tail, reducing it in number by 2 carbons. So let's see if we can follow this through one 2 carbon snip. And we did this a little bit before, but again, it's important that we get a sense of how much ATP energy is produced per 2 carbon snip. Well, the SNP itself, taking those two carbons, those initial two carbons that are going to become acetyl-CoA, removed from it, that process drives the formation, let me clean this up a little bit, drives the formation of one FADH2 and one NADH just by snipping two carbons from that chain, which ultimately ends up as acetyl-CoA. So that acetyl-CoA, as we know, can then be put into to drive one turn of the citric acid cycle. This acetyl-CoA now, we see, this is something we've looked at before in the previous unit. As we drive this one cycle, this one turn of the citric acid cycle, we're producing a lot of NADH, a lot of NADH, some FADH2, and we are driving directly the formation of one ATP. One ATP. Remember, the citric acid cycle itself, to directly produce ATP is ATP poor. Where you really get all the ATP is in the formation of these ATP equivalents, NADH and FADH2, which are cashed in later for driving the formation of a lot of ATP through oxidative phosphorylation. But again, we made a rough count last time. Per SNP, we get 5 ATP. Per 2-carbon acetyl-CoA that is produced from each SNP, we get 12 ATP. So let's look at how this will work itself out on a representative fatty acid. We'll try to do a rendering of the total amount of ATP that we get from a representative fatty acid. So a representative fatty acid that we'll look at is known as stearic acid. That is 18 carbons long. It doesn't have to be 18 carbons. We could look at other ones, 16 carbons, 20 carbons, or whatever, and go through the same process. But this is just to show how this is ultimately going to work out. Now, if we look at our stearic acid here, we can see that this can be broken down. These 18 carbons can be broken down, and this is inline drawing, into 2-carbon fragments, and here they are. I'm just showing them 2 carbons at a time. We find that if we could get a total of 9 2-carbon fragments out of it by snipping it a total of 8 times. So here we go, and this is very crude, and we'll look at this in a much better, more artistic fashion as we move along. But we would have 8 SNPs giving 9 fragments. Now again, the term SNP is not a scientific term, but let's look what happens if we more artistically cleave this 18-carbon stearic acid into 2-carbon fragments. Well, we ultimately, if we look over up on the top part here, we see that we get a total of 8 SNPs, 8 2-carbon fragments. And let's look very carefully here now. We get a total of 8 SNPs giving 9 2-carbon fragments. So that is ultimately where we're going to start with. Remember, each time we snip 2 carbons off, that drives the formation of 5 ATP. Each 2-carbon fragment powers 1 turn of the citric acid cycle, which is in essence 12 ATP. So let's add all this up. And we'll summarize. So we have 8 SNPs, 9 2-carbon fragments. As we said, each snipping event produces 1 FADH2 and 1 NADH. If we add all those up, that totals 8 FADH2 and 8 NADH, all of which can be cashed in for ATP. Now, those 9 2-carbon fragments are all essentially acetyl-CoA. So they can be in the form of acetyl-CoA. That's going to power 9 turns of the citric acid cycle. So I'll clean this up a little bit, and let's see where we end up here, if we can sort of give a rendering. Remember, in the very beginning, we have to spend our own ATP, 2 ATP, to activate this stearic acid and to hook it up with the coenzyme A. So that initial energy investment is minus 2 ATP to start us off. Well, we get 9 2-carbon fragments, which are essentially 9 acetyl-CoAs produced. Now, each of those acetyl-CoAs powers one turn of the citric acid cycle, which gives us net 12 ATP. So that immediately, now those 9 acetyl-CoAs give us 108 ATP. And that's a lot. Remember, each snipping event, we had 8 snipping events, snipping 2 carbons off at a time, gives us 5 ATP per 2-carbon snip. So adding that up gives us 40 ATP. And if we add all this up and take back out the initial 2 ATP investment, we end up with a remarkable 146 ATP per 18-carbon stearic acid. And we find that fats in the form of fatty acids are much more energy dense, say, than other fuel molecules such as glucose. But this it doesn't really matter that we're doing this on stearic acid. If it happened to be we're just happening to do it on stearic acid with 18 carbons, we could do it just as easily on a 20-carbon representative fatty acid or the 14-carbon representative fatty acid. It would all be the same. We would still come up with the same sort of rendering we have here. We would just change up the numbers a little bit.